Right folks, this is the final video for the Higher Human Biology course. It's the last part of the neurobiology topic. I wouldn't expect you to be watching this video unless you have done the first of the two neurobiology homeworks, so make sure you've done that homework, which was covered by those 3 videos, before you start thinking about doing this one. This covers quite a lot. It's going to be a reasonably long video. In that situation, as I've told you before, I expect you to pause it, wait until you've taken something and maybe watch it a couple of times and then watch the next bit. What I'm going to attempt to do in order to let you guide yourself through this video is when I'm on each of these individual topics, it's kind of 5 sections. Each of those sections I will cross out once I've done, so you'll be able to, by just looking at this list, be able to tell what stage we're at each point, okay? So just to save any more time, because we're already a minute in and I haven't actually told you anything, we're going to start making our way through this topic. Now there's one homework remaining that covers all of this information. That would be what I would expect you to do very last. It's currently the first week of the Easter holidays and I've set you the first of the neurobiology homeworks for next Wednesday. The idea is I would set the next homework for basically the Wednesday for the following week, the Wednesday that you come back. So that's how it's all going to work. Hopefully that makes sense. You may see me looking down now and again because I just want to double check I don't forget anything, because there's quite a lot of information in this topic. So without any further ado, we'll crack on with the first structure, the first section, the structure and function of neurons. So at National 5 level, you learned what neurons were. They're basically nerve cells, another name for nerve cells. They come in different types. What their job is to do is to carry nerve impulses around the body. They will carry information through a nerve in the form of electrical impulses, but between nerve cells, if you remember, they transmit a chemical, that chemical called neurotransmitter, and we'll talk about that in a wee minute. But the main sort of thing you need to understand for this first topic here, the structure and function of neurons, is there are basically 3 main parts of a neuron. Okay, so you've got the sort of part you might recognise as being the cell. It's got its nucleus, it's got cell membrane, it's got a bit of cytoplasm. Most nerve cells have quite a long structure that carries the impulse along them. Draw something around that, which I'll name in a wee minute. And then at the end, we have, quite often, a nerve cell has several places where the nerve impulse can go. So that's your basic standard nerve cell, or neuron. To label the parts, this is called the nerve cell body. That is called the axon. These are called the axon terminals. And what the nerve cell body is receiving information from other cells, from other axon terminals basically. Those parts there that receive the information are called dendrites. So you have information being passed from a nerve cell to this one, received by the dendrites. The nerve impulse then travels through the cell, down the axon, till it reaches the end of the axon terminals, and then that signal, that impulse is then, under certain conditions, it's then passed on to the next dendrite, and that's how information is passed on. You'll remember in my previous video I had told you that an electrical impulse passes through the cell, but it's a chemical that's passed between cells. Okay, and we're going to talk about what happens once an impulse reaches the end of a nerve cell and what happens in order for it to get to the next one in this second section. But the main information you need is really on the board at the moment. The only other thing to mention is this structure around the axon that I'm shading in orange here. The axon's the long structure that comes away from the nerve cell body, but this orange stuff that I've coloured in has a special name. It's called the Myelin Sheath. And the Myelin Sheath's job is to increase the speed of nerve transmission down the axon. It insulates it basically, so there's any electrical impulse, it passes from there to there quicker. Myelination is the process of a cell developing a myelin sheath around its axon, and what you'll generally find is that very young children have cells which are relatively unmyelinated, which basically means they've got less myelin than an adult, and that's why small children are slightly less coordinated than adults in terms of being able to move, being able to walk, being able to get up and catch a ball and all these other things that you know adults are generally better than children at. It's because of the myelin sheath there. Okay, so those are the structures of the nerve, an ordinary neuron. Occasionally, depending on what kind of neuron it is, because you possibly remember from National 5, there are different kinds of neuron. We get motor neurons for carrying signals from the brain to muscles and glands. We have sensory neurons that carry signals from the sense organs like the eyes, ears, nose, mouth to the brain. We also have interneurons, which are found inside the spinal cord to connect directly a sensory neuron with a motor neuron to allow reflex actions. So depending on what type of neuron it is, you might find that the setup is slightly different. You might find that the dendrites are found on the end of the axon and the nerve cell body is actually found in the middle. It ultimately doesn't matter. You need to be able to identify the main parts of a neuron, whatever kind of neuron it is. They're all the same basic structure. Myelination. The other thing to mention about this myelin sheath, which I nearly forgot there, as I said, I might now and again check down just to double check I'm saying everything that I need to. You will get some diseases that affect can break myelin down and reduce the amount of the myelin sheath that there is. And if you've got a disease that does that, you find you will start to lose coordination. You don't need to know the types of disease, you don't need to name them, just be aware that there are diseases which can reduce the amount of myelin on an axon, and the knock-on effect of that is that the sufferer of that disease might lose coordination, etc. So that's really the end of the first topic, the structure and function of neurons. And that's the structure there, basic structure. Now obviously if you feel I'm going too fast, then you can just rewind me. That's not what you get to do in class, but that's what you need to do if you feel I've moved through that too quickly. Remember, of course, you'll have your notes to go with this as well. They can be found in the files section in the higher team. Right, on to neurotransmitters at synapses. What I'm going to start off by drawing is a very close up. I'm going to draw an axon terminal, so that's the very end of the nerve cell. So we have an impulse, potentially an impulse being sent down, and this is the end of the nerve. And underneath that, I'm going to draw the dendrite of another nerve cell. Right, so basically the direction of the impulse is that way, right? It's going from that nerve cell to that one, but what you'll hopefully notice is this space. This space at National 5 we talked about being called the synapse. In actual fact we talk about it now as being the synaptic cleft, but ultimately it's the same thing. There is a term used to describe the nerve before the synapse and the nerve after a synapse as well. This one is called the pre-synaptic nerve, for obvious reasons. If it's not obvious, because it comes before the synapse. And this nerve, with the same idea behind that one, be called the pre-synaptic nerve. Can you guess? Post-synaptic nerve, right? So post-synaptic nerve. So that's the sort of terminology. That's to try and give you to zoom right in and understand what we're looking at here. We are just spelled impulse wrong as well. Probably noticed that already. Then the nerve impulse comes through a nerve, it gets to the end of the presynaptic nerve, and somehow the signal has to get from here to the postsynaptic nerve. It has to get across this space. And because we're talking about electrical impulses, electrical impulses can't jump across that space. So something has to happen to that nerve cell to allow the signal to be passed to that one. And it absolutely does. That's exactly what happens. We use a chemical called neurotransmitter. Now, neurotransmitters are stored in vesicles in the axon terminal, right? So we're going to use a red triangle to denote neurotransmitter. So your red triangles are chemicals called neurotransmitters, which spend their time at the moment stored up in vesicles inside the axon terminal, inside the presynaptic nerve, just waiting for the instruction. We'll talk about what that instruction will be in a minute. So they're just sitting there waiting. If we get an impulse coming down from, you know, let's say this is a sensory neuron, so there's a bit of pain somewhere in the body and it's been sent, and we're needing to get back to the brain to say something's sore, so do something about it, stop the pain, somehow behave in such a way where you'll stop the pain. So if that impulse does reach these vesicles, the trigger for the vesicles to then move towards the axon terminal, so they'll move towards the membrane. And vesicles, as you know, we've met vesicles before, this idea of a sort of circle of membrane, and membranes can fuse with each other, so that's exactly what happens. This neurotransmitter that's stored inside the vesicle makes its way to the membrane, fuses with the membrane, so the membrane becomes a sort of continuous structure. And as you can hopefully see, the neurotransmitter is then free to move out and into the synaptic cleft. Now, if there's enough of an impulse, that will happen to enough of them. The sufficient number of vesicles will fuse with the membrane until we actually have enough neurotransmitter inside the synaptic cleft here. And when I say enough, it's because there is a threshold of neurotransmitter where if you're below that threshold, you're not going to get the signal passed on. We're still not really explained how the information actually gets passed on. All we've really explained now is that if there's an impulse in the presynaptic nerve, that will be enough to release some neurotransmitter into the synaptic cleft, but doesn't really explain how this cell is able to react to that. You might already be a one step ahead because we have talked about some, similar ideas in biology, but you've got a signalling molecule here. If you want that signalling molecule to cause a reaction in this cell, then this cell is going to have receptors for that structure. So all along the dendrites, you will find specific receptor proteins which will fit the neurotransmitter. So it will be released into the synaptic cleft, it diffuses across the synapse and then starts to fill up receptor sites, receptor proteins on the surface. Now, there'll be, as I said, a threshold. You might not need every single one of those receptors to be filled. You might though. That doesn't really matter, but as long as you understand the concept that there's a threshold number of neurotransmitters, if they are bound to receptors, then that will basically cause that impulse to be sent on. If you go on to do advanced higher, we will learn a lot more about how that actually does it. But at the moment, all we need to understand is that if we have enough neurotransmitter bound to these receptors, then that impulse that was previously getting passed down that nerve will then start to get passed down that nerve, only for the whole story to repeat. Remember that all the way through the body, you have several, like, you know, a chain of many nerve cells to get from one place to any other place. And it's every time you get to the end of a nerve cell, you're going to get to a synaptic cleft. So this neurotransmitter has to get released into the synaptic cleft and then bind with receptor proteins for the impulse then to be passed on to the next cell, and for that impulse to keep going through the body. Now, something that is obviously going to be required is for this neurotransmitter at some point to go away. Because if you think about it logically, if this neurotransmitter was just to stay there for the rest of the time, then whatever this impulse was, whether it was pain or whether it was blue light or whether it was a bad smell or whatever information it happens to be, or whether it was clench your fist or bend your arm or raise your knee, if that signal has been sent to that cell and this neurotransmitter never goes away, then that signal is just going to get sent forever. And obviously you don't want that. You don't want to continuously send a signal once there's no need to send it anymore. So the next thing to talk about in this topic, and this part of the topic, is the idea that this neurotransmitter has to go away. And there's two different ways it can go away. Firstly, it can be reabsorbed back into vesicles. So that's one way that you can deal with removing the neurotransmitter when you no longer need that impulse. So you can reabsorb neurotransmitter, or another thing that can happen is, along with these neurotransmitters in the vesicles, you will also find enzymes which are specific for breaking this neurotransmitter down. So if you release an enzyme into the synaptic cleft that's job is to break the neurotransmitter down, then eventually what happens is, well, that's exactly what happens. The enzyme breaks all the neurotransmitter down and you no longer have any neurotransmitter. Now, depending on what kind of neurotransmitter it is, some of them are reabsorbed, some of them are digested by these enzymes, but ultimately it doesn't matter. You just need to understand there are two different ways of neurotransmitter being removed from a synaptic cleft. You don't want, as soon as this neurotransmitter has been released, you're more or less looking for a way to get it out. You're more or less looking for a way to eliminate it from the synaptic cleft. Now, bear in mind as well that neurotransmitters can have either an excitatory effect or an inhibitory effect. Right? But by that, what that means is that some of these cells will be switched on if neurotransmitter binds, some of them will be switched off if neurotransmitter binds. It's important to understand that the process of a neurotransmitter binding to a receptor can either have a positive effect on this impulse being sent, or a negative effect. Okay, very important to understand that there are those two different effects. The way a synapse is designed as well, a synaptic cleft, it has multiple neurotransmitters. What that means is that very often, see if there's not enough of a signal sent through, if there's not enough of these, then if there's not enough neurotransmitter bound to these receptors, there will be too weak a signal, and because the signal's too weak, the actual impulse won't be sent through the neuron. So that's another thing to understand as well. However, you will hopefully remember from the previous video I talked about converging neural pathways. Now that's something to consider at the moment, because let's say that one nerve sending a signal, releasing its neurotransmitter is not enough to trigger a response, but try and imagine you've got several axon terminals all feeding in the same way that I've described already that a converging neural pathway will do. So if you've got several cells all releasing neurotransmitter, then you might actually reach the threshold you need to get a signal, right? And remember, that's the places like, for example, your body's getting different information from all over the body in order to send a signal somewhere. So if a number of different neurons are all feeding into the one dendrite, then that can actually lead to there being a sufficient response. So several weak neurotransmitter responses can actually collate together. It's called, what is it called? It's called summation. Process of summation, where all these neurotransmitters gather together and you get just enough to cause a response. Okay, so that's it. The only thing, I forgot to mention as well, actually, see when we go back to here, I'm going to write it up here just as a reminder because this will be important for the first topic. Glial cells, don't think I mentioned these. Glial cells support the neurons and also they produce the myelin sheath. Just suddenly popped into my mind that we need to talk about that. Okay, so that's part of that first topic, glial cells. Right, so that's the fundamental idea behind neurotransmitters at synapses, so we can kind of put a line under that one as well. I think that's probably the two real meaty bits. I think these ones are reasonably straightforward. We're at 21 minutes, so I think what I'm going to do is upload this as 2 videos. So this is the first of the 2 videos that I need for you to watch before you try the final homework. So what we've covered then is the structure and function of neurons, including a bit about glial cells just at the end of the video there, and we've also looked at the idea of what happens actually at a synaptic cleft. Neurotransmitters released from these vesicles diffuse across the synaptic cleft and bind with receptors. They're then removed either by being reabsorbed back into the nerve cell or by being broken down by enzymes so that we don't get this sustained response. Remember, the response can either be excitatory or inhibitory, and if only a small number of neurotransmitter gets released, sometimes the response can be too weak to be passed on, but that can be increased through the process of summation when a number of different nerve cells in a converging pathway come together, all releasing their own neurotransmitter to bind and give sufficient neurotransmitter to actually cause an impulse. Okay, so we'll call that video 1. It'll be the same screen, etc. Start a video 2, but I'll take that drawing away and we'll start off by talking about neurotransmitters and how they can affect mood and behaviour. Okay.